Hello, my name is Kathy Hernandez, and this is my presentation on the research article, The Screening of Apoptosis Pathway-Mediated Antiproliferative Activity of the Phytochemical Compound Virinosdinon against Human Non-Small Lung Cancer A549 cells. So the phytochemical compound virinosdinon is considered an anti-cancer agent against A549 lung cancer cells in this study. To the right, we have the structure. This research study investigated the phytochemical compound and it highlights the cytotoxic effects of FDN and the molecular mechanisms that play a role in inducing apoptosis and cell cycle arrest. So this study is important in understanding how the phytochemicals have a potential as an alternative to traditional chemotherapeutic agents with lower toxicity. So figures 1 to 7 and 8 were the figures of most importance that we're looking at. So figure 1 observed the FDN effectively targeting A549 cancer cells, while figure 2 showed that the FDN induces apoptosis, which are important in treating lung cancer cells. So in figure 1, the techniques used were cell viability assays. The cell viability assay that was used in this specific figure is MTT assay. This measures the ability of living cells to reduce a yellow dye, MTT, to purple for Mazan crystals. The reduction occurs only in a metabolically active cell. So this allows researchers to assess cell viability based on color intensity. The principle behind this technique is that more viable cells presence means more formazan produced, leading to a higher optical density measured in the spectrophotometer. So in the methodology of the MTT assay, 549 lung cancer cells were cultured and treated with varying concentrations of FDN, ranging from 0 to 300 micrometers. So the concentrations that killed half of the cancer cells were IC50s, and that was about 85 micrometers, micromolars. The cell viability in Figure 1A showed that the concentrations 50, 100, and 200 micromolars showed FDN significantly reducing cell viability. You can tell by the significance by the number of stars. One is the most significant. This is comparing the control and 50 micromolars of FDN. And then the three stars means that those two are comparably the same significantly. And so the 200 micromolar dose on this part 1A showed that the cytotoxic toxic effect was similar to a standard chemotherapy drug cisplatin. So you can see that in the far right side of 1A, the last two bars is 200 micromolars FDN and 200 micromolars cisplatin, and they're almost significantly the same. So in figure 1C, we can see the morphological changes that were observed under a phase contrast microscope. This showed that the A549 cells that were treated with the FDN displayed less cell confluence and had signs of apoptosis, such as blebbing in the plasma membrane. And then additionally, the FDN treatment caused many cancer cells to detach from the surface while untreated cells remained attached. So these results could contribute to answering the main research question of how these phytochemicals can be used as therapeutic drugs for lung cancer. So one can be that the significant reduction in cell viability at various FDN concentration indicates that there are potent cytotoxic effects specifically against A549 cells that support the hypothesis that FDN could serve as a potential anti-cancer agent. So the selectivity also is important because the cytotoxicity towards non-cancerous J774A1 cells suggests that FDN selectively targets cancer cells, which is a vital characteristic for any potential cancer treatment. And then in figure 2, the technique used was the host 33342 staining, and then annexin VFITCPI assay. So the Host 33342 staining is a technique that is utilized for using a fluorescent dye that binds to DNA. When the cell undergoes apoptosis, chromatin condenses and leads to an increase in binding of the dye, resulting in a brighter inflorescence. So we can see that in part A, the inflorescence and the different quadrants. So the top left is the control, and then the different concentrations of FDN treating these A549 cells. So the A549 cells treated with FDN showed a brighter blue fluorescence after staining with the host 33342 compared to untreated cells, especially at higher concentrations. The increased fluorescence indicates that a greater chromatin condensation a sign that apoptosis is occurring. So in order to quantify these fluorescence, the intensity of the blue fluorescence was quantitatively measured, and the FDN-treated cells exhibited higher fluorescence intensity than the control cells. So in order for the methodology of the host staining procedure, A549 cells were treated with varying concentrations of FDN, and then the dye was added to the cells, and then they were visualized under a fluorescence microscope. So for the annexin VFITCPI assay, the annex leafy Figure 2C, this assay was used to quantify the apoptosis. Cells in the lower right quadrant indicated late-stage apoptosis, while the upper right quadrant is the represented early-stage apoptotic cells. This flow cytometry-based assay is able to distinguish the viable early apoptotic and late apoptotic cells. Annexin B binds to the phosphatidylserine, which is translocated to the outer leaflet of the cell membrane during early apoptosis. So propidium iodide, PI, stains the cells with compromised membranes, marking them as late apoptotic or necrotic. So these contribute to answering the overall research question by finding the assays providing compelling evidence that FDN induces apoptosis in A549 lung cancer cells. So the staining results and the fluorescent intensity measurements indicate that FDN treatment leads to chromatin condensation, and the Annexin B FITCPI assay results confirm that FDN effectively promotes both early and late stage apoptosis in dose-dependent manners. So we can see that the control is the brightest on the lower left, and we're most importantly looking at the upper right and the lower right. So the ones with the darkest are the 100 micromolars and the 50 micromolars. For figure 7, the techniques used were flow cytometry and quantitative qPCR. And the technique used for quantifying a population of A549 cells in different phases of the cell cycle was found using flow cytometry. This method relies on the principle that cells can be stained with DNA binding dyes, such as the PI in the previous slide, which fluoresces in proportion to the amount of DNA present in the cell. As cells pass through the laser beam and flow cytometer, their fluorescence is measured and allowing for determination of distribution of cells across different phases of the cell cycle. So we're looking at G0, G1, S, and G2M. So the methodology around this is that the cells were treated with varying concentrations of FDN, such as 50 micromolars, 100 micromolars, and 200 micromolars for a duration of 24 hours. Then they were stained with the DNA binding aid dye to allow for the measurement of DNA content. Then the flow cytometry analysis, the stained cells were passed through a flow cytometer and they were measured by fluorescent intensity of each cell. So figure 7 presents the results of the flow cytometry analysis, and they showed that a percentage of A549 cells in the G0, G1 phase after treatment with different concentrations of FD9. So the data indicates that a dose-dependent increase in the percentage of cells in the G0, G1 phase. So we're looking at the G0, G1 phase for 50 micromolars, 100 micromolars, and 200 micromolars, and they're higher than the control, meaning that there are more cells in the G0, G1 than in the control. This means that FDN treatments lead to significant alterations in cell cycle distribution. So the accumulation of cells in G0, G1 phase suggests that FDN inhibits cell proliferation, which is a key mechanism in the induction of growth, inhibition, and apoptosis. And then the B on figure 7 is just a bar graph with the quantitative of the left side. In figure 8, the technique used was flow cytometry and the quantitative PCR as well for gene expression. So for flow cytometry, this technique relies on the principle that cells can be stained, as said previously, and it's determining the proportion of cells in different phases of the cell cycle. So we're seeing more cells in of this figure 8. So in figure 8, we're looking at the gene expression analysis, and then it illustrates a change in gene expression levels of cyclin D1, CDK4, and P21CIP1 after FDN treatment. So these are the different uh, fold change and the relative to the control. So we're seeing a more significant in the control and 50 micromolars for CDK4 with the 1 star. And then we're seeing a more significant with the 50 micromolars compared to the control in the P21 CIP1. And we're also seeing somewhat of a steady control versus other concentration of 50 micromolars to not be as significant as the others. So there's a decrease in cyclin D1 and CDK4 and an increase in P21 CIP1. This supports that the conclusion that FDN disrupts the regulatory mechanisms governing cell cycle progression. So in figure 1, it illustrates that FDN effectively targets A549 lung cancer cells while sparing normal cells, and it induces characteristics typical of cell death. In figure 2, there shows evidence that FDN induces apoptosis in A549 lung cancer cells, and figure 7 shows that the accumulation of A549 cells in the G0 and G1 phase upon FDN treatment supports the hypothesis that FDN inhibits cell proliferation. Figure 8 shows how FDN disrupts the regulatory mechanisms governing cell cycle progression, thus contributing to the overall understanding of its effects on F549 cell growth and apoptosis. So overall, FDN has a multifaceted role in targeting A549 lung cancer cells. It is selectively induces apoptosis, inhibits proliferation, and disrupts cell cycle regulation. FDN demonstrates the potential as a therapeutic agent in lung cancer treatment.